Hello, Assalam Alaikum. Welcome to another video. In this tutorial of MCAT, we are going to study about the life cycle of a bacteriophage. What is a bacteriophage? It is another name for the viruses of bacteria, which infect bacteria. But it's not really relatable that the cycles we are going to study might be applying to the viruses of the animals as well. So let's get started and into the depth of the topic. So today our video is going to be of about 10 minutes inshallah, because I have added a timer. So if I speak out a lot, then the timer is going to just turn off by itself. So that's what's going to make my work very hard, and it will make me go as fast as possible. So first of all, we have two types of cycle. One is lytic cycle and the other is lysogenic cycle. These both of these cycles are the cycles for the reproduction of the virus. A virus undergoes its reproduction in its host cell. So let's have a look at the lytic cycle. Lytic cycle is a type of cycle in which the host cell bursts out for the reproduction of a virus. It is a viral reproductive cycle that cul- culminates in death of the host cell is known as a lytic cycle. What a typical definition and very difficult wording. So it's just trying to explain you that in this method of uh, reproduction, there is death of the host cell. Lytic cycle can destroy the entire bacterial colony in few hours. As we have experimented the viruses over the bacterial colony and bacterias, that's why it's explaining the timing of a virus to destroy a bacterial colony. We do not consider animals because the study has been done on the bacterias. Viruses were thrown to the bacteria and a bacterial colony bursts out within few hours. Viruses with lytic reproduction method are very virulent, as we know. The host cell is destroyed. Phases of lytic cycle. Now, this is a step-by-step procedure by which a lytic cycle attacks a cell. First of all, the T phage uses its tail fiber to stick to specific receptor sites on the outer surface of an E. coli cell. E. coli cell is a cell type of cell which we have studied this phase over, and the T phage is the name for the phage or name for the virus. As I told you, phage is being used for viruses. So it just stick its tail fiber. The sheath of the tail contracts, thrusting a whole core through the wall and membrane of the cell. It contracts, it just comes over the cell membrane and then contracts like this. You can see my hand, and these are the tail fibers, which are my finger projections, and it contracts like this. And after that, we have that it injects its DNA into the cell. After that, we studied that there were some sticks coming from over here in the virus, which will go into the cell membrane. uh, I I don't think so. You might be hearing me properly, or maybe you would be. So just have a look at it. There is another pin coming out of it which enters the membrane and then inject its own DNA. The empty capsid of the phage is left as a ghost. Outside the cell, the cell's DNA is hydrolyzed. Now, what happens? The empty capsid, the DNA containing portion of the virus is known as capsid. Inside it, there was DNA. It injected its DNA into the cell. Now it is empty and it's just like a ghost. It has no work, it is useless. The cell's metabolic machinery, directed by phage DNA, then produces phage protein and nucleic and DNA and makes it copies, which means now the DNA injected inside the cell plays its role and makes up new DNA for a new phage, because phage has to reproduce, so it makes protein and DNA for the phage, which is virus, and makes its copies. These phages then direct production of lysozyme an enzyme that digests the bacterial cell wall. Now this, the phage which are being produced and which is over the cell, all produce lysozyme. Now these lysozyme do, they just destroy the bacterial cell wall, or whatever the host cell wall we can say, because it can be an animal cell. So it destroys the host cell wall, and which in turn there is osmosis, and all the extracellular fluid enter the cell, and cell swells up and then just bursts. And after the burst of the cell, we get 100 to 200 phage particles. This is how it done uh, visually. You can see in the first picture there is a bacteriophage sticking to the cell, then it induces or inject its own DNA. After it, we have so many viruses inside the cell. Now bacteriophage, there is, now it will release the lysosome enzyme to the membrane and then burst it out, and the cell dead. The cell is dead while we have so many viruses. After it, we have a lysogenic cycle, reproduction without destroying host cell. In this type of cycle, the phage comes over the cell, injects its DNA. As you can see in the picture, phage attaches to the cell surface of bacterium, and then enters its DNA into it. Phage DNA enters the bacterial cell. Now prophage starts the phage DNA integrates into bacterial DNA. Now in this type of cycle, the DNA of the virus is kept inside the cell, bacterial cell, it just go fix into their own cell, their own DNA. Bacterial DNA and virus DNA both merge and they become one. And after that, when the bacterial cell reproduce, they form two daughter cells. Now these daughter cells have been affected by one virus. Now these virus can activate anytime. Now this DNA inside these cell is preserved whenever they want, whenever they are triggered. It's not been determined in how they are triggered, but they can trigger and start producing, just like in the lytic cycle. As we see, the DNA is injected. Now that part is being done in lysogenic phase. The DNA has been entered. Now this DNA is inside the bacterial cell living, and it's not performing, it's sleeping, we can say. But whenever it get, at any moment when the virus is triggered, the viral genome detaches from the host cell's DNA and enters stage 2 of lytic cycle. And what happens in the stage 2 of lytic cycle? The we can say the phage DNA start producing its own head, tail piece, and the sheath and everything, and start making its copies. And then in the end, death of the cell occur, and there are 100 to 200 phage particles. That's what it is. In short, lysogenic cycle is a preservation of the cycle of the lytic cycle, and lysogenic cycle also follows the lytic cycle. In the end, whenever the trigger, the the viral DNA is triggered, and this triggered has not been discovered yet, and inshallah maybe it will be discovered in future. So thank you so much for watching. This was all today's lecture. Like, give a thumbs up, and subscribe for more videos, and subscribe to get notification of more MCAT videos which I am making soon enough, and I am trying my best to regularly update the videos of MCAT, because I know we are going to have our test from 15th September to 15th October. Share with your friends. Believe me, your friend is not going to take higher marks from you. Just go ahead and share the link in your WhatsApp group where you have many medical friends. It will help me stay promoted and reach 1000 subscribers as soon as possible. Suggest me any improvement you think I should do, and any topic if you want to be covered by me, just mention it below if you want it to be done as soon as possible. And if you have any questions about the topic, it will be entertained, and I am online as much as possible, so I will be very quick to reply to your queries, so you can comment below any question you have. Allah Hafiz. Thank you so much for watching.